All right. Now we're at the last portion or the last part of this workshop on spatial phylogenetics, where the focus here will be to discuss what the conservation applications of using spatial phylogenetics. But beyond that, also talk broadly about even the empirical uses of spatial phylogenetics, the various areas and various research fields where you can apply spatial phylogenetics. So I want to start by saying that you can use these methods. I'm sure you agree with me from what we have discussed so far in the last 6 parts that spatial phylogenetics can be applied to answer questions in various biological fields, focusing, for example, biogeography. Biogeography is a field that looks into describing patterns of biodiversity in time and space. And so I think spatial phylogenetics fits perfectly with this area. We can use these methods also to answer questions or to elucidate on concepts of evolution. For example, Brent had already mentioned concepts like refugia, areas that were refugia during the glaciation periods, diversification, rates of diversification, which regions have experienced those versus other areas. And so we can use these methods to elucidate on that. We can also answer questions on ecology here, especially when we talk about interpreting high and low phylogenetic diversity. You can also think of it in terms of phylogenetic overdispersion or clustering areas. Clustering would be areas where there's high concentrations of lineages that are closely related to each other versus areas where they are phylogenetically distantly related from each other, representing overdispersion. And then we can begin to ask questions like what ecological processes are responsible for these kind of patterns we see on the landscape. And then, of course, as we've already been discussing in terms of conservation, we can shift the paradigm of looking into using spatial phylogenetics to answer questions on which areas need to be conserved right now, especially given the fact that habitat destruction is still the greatest threat to biodiversity. And now we have the global initiative, which is called 30 by 30, that is protecting 30% of lands of the Earth's terrestrial and marine or aquatic habitats by the year 2030, something which the current US administration has bought into by developing what is called the America the Beautiful initiative, called 30 by 30 initiative. That is protecting the 30% of US land and water by the year 2030, and something which California has also bought into. We have California 30 by 30 initiative as well. So instead of selecting areas given some charismatic species or species richness, we can actually use spatial phylogenetics to determine which areas need to be protected given what we have already protected. And that is one of the things that we're going to be talking about in this chapter. So how do we select new areas for protections, considering the fact that we have existing protected areas? If you go, we have the world database of protected areas, which shows you which areas are protected in the world, and you can be able to look into that. And of course, we all know that there are also different degrees of protection. You will not compare, you will not place BLM protected lands on the same level with national park, for example, or a wilderness, because there's a lot of anthropogenic activities that are still being permitted on BLM lands versus in a national park. So then we have to also think about those as well. So this is like a general framework which was discussed, developed as part of the California-funded California Plant Phylodiversity Project, part of which has already been published in 2017, 2018, looking into which areas need prioritization for protection or conservation using spatial phylogenetics. So let me walk you through this, framework of protocols. So we start with the occurrence records, which are generated from the online databases or herbarium records. And then we model their range, given that we don't have a full understanding of the occurrences of each of the Californian taxa. So we model their range using climatic conditions. And from that, we get the distribution models. And then we combine the distribution models with the phylogenetic tree for all taxa that we have data for in California. But then we're not just predicting their ranges based on the favorable climatic conditions. And so we don't want to be predicting suitability, high suitability or high probability of occurrence for a taxa in the middle of Berkeley or in the middle of San Francisco or Oakland, where those areas are already fully built up. So then we integrate, or we bring in what is called landscape intactness, which shows which areas in California are still pristine, are yet to have major anthropogenic alterations on the landscape. So we bring that in as a layer, and then we also bring in protected status or protection status, which shows us which areas in California are already protected. And so that we can be able to, after doing all of the analysis, we can be able to say, well, these areas are already protected. These areas are not yet protected, but they contain unique biodiversity that needs to be protected. And so, again, bringing back into the protection status map, what I mentioned earlier, that different protected areas have different degrees of protection or management. And so the authors of this study, what they did was that they gave different ranking to different protected protection status of different lands. For example, national parks and wilderness are ranked higher compared to BLM lands or some other areas, given the different management practices on those lands. And then, so we now have a phylogenetic tree. We have a phylogenetic tree. We have distribution models predicting, which predicts the range of all the taxa in California that was used for this study. And then we have this layer of protected status or protection status, which are the three entities or three components of that model. So what was done is the phylogenetic tree was combined or integrated with the distribution models to produce those spatial phylogenetics, those metrics that Brent had already discussed in previous parts in terms of the phylogenetic diversity, the phylogenetic endemism, the relative phylogenetic diversity and endemism, and all the other metrics were computed using the tree and those models. And then, in addition to the protection status layer, they were now brought into a different program. And what that program does is a stepwise algorithm that helps us to compute. Let's assume this one particular grid cell is computed in spatial phylogenetics to have unique high phylogenetic diversity, for example, then we want to be sure that part of that range or a greater portion of that range is not already protected in existing protected areas. If so, then the model scores that grid low or ranks it a little bit lower because that that particular lineage or the branch lengths are already protected in existing protected areas. So basically what that does is that you are giving serious considerations for the occurrence of the taxa, the branch length for each of those taxa, and then their range size, the part of the range size which are already protected or not yet protected in the protected areas, and then, of course, the portion of the range that is already protected and the current protected status, which is what I already explained. And then the representation of those taxa across the proposed reserve network. So let's say one grid cell is selected as meeting all these requirements such that it has high unique evolutionary attributes or branch lengths, a higher PD, for example, and the range size or portion of the range size is not already protected in existing protected areas, then that grid cell is scored or is selected. And then that algorithm goes back again and rerun the entire analysis, given that already selected grid cell, which other aspects or areas of the study area needs to be protected. And then it continues to select it in that iterative mode until the entire model becomes stable. In fact, when I ran it on my computer, on my local machine, for more than 2 weeks, it was still running to tell you how much of the iterative process it goes through. So this is a stepwise optimization algorithm that was developed as part of this funded project, of which Brent was one of the coordinating PIs, whereby it computes or ranks important areas that need to be given prioritization as areas which are poorly protected. Areas that are not yet represented, that is, the range of those taxa are not found in current protected areas in California, and they have long branch length, branches or branch length, small range or poor range protection. That is, a portion of the range is not protected. Portion of the branch lengths are not yet protected in the existing protected areas. And it also complements, that is, we're not having redundancy. We want to avoid redundancy such that areas that are being recommended for protection are not already protected in the existing protected area. That is basically what we're trying to avoid in this study, in this kind of analysis. And so again, for example, this is an example of the map of the protected areas in California, and we can see that areas that are already protected are found around the southern portion of the Sierra Nevada, for example, the Death Valley National Park and other protected areas are along that area are already protected. So areas in yellow have current known protection status. And I also want to say that, especially given the explanations have been given earlier by Brent, that this analysis can be done with any of the facets of phylodiversity. For example, we can do that with the chronogram, as already been explained earlier. And in this case, we want to give preference to taxa that are older lineages, so they have accumulated ecological experience over time. And so even though we may have areas where we have ginkgos or magnolias, and maybe they are not really they are not really species rich, but those taxa are older lineages, which needs to be, which are given priorities if we are using a chronogram, for example. And then if we want to, we might want to use a phylogram for the analysis, because we want to think about which lineages have the greatest rate of divergences, or the net divergence. And so we want to think in terms of the fact that these lineages, if they have higher divergence versus other lineages, then we might want to be thinking in terms of the fact that those lineages have some evolutionary uniqueness to them that are important for maintaining biodiversity, or post-climate change apocalyptic situation. These are lineages that could result in developing or evolving new biodiversity in those kind of hypothetical situations. And then, of course, the same thing applies to cladogram, whereby we want to think about giving priorities to lineages that are more specious, that is, they have more than random rates of developing new lineages or new taxa than others. And so we want to give priorities to those lineages. So these are different questions or different important aspects of biodiversity that we want to protect using different facets, which has already been explained by Brent earlier. And so again, so running that analysis, this was what was this was a result. And so I'm going to walk you through this result or the map. So first, looking at the map of California itself, you can see that there are some areas that are there are shades of gray on that map. And so areas that have dark gray color are areas that are already protected. And areas that have lighter shades are areas that are not currently protected. And so those were ranked to be more important than areas that are already protected. And then the analysis was conducted with each of the three different facets individually, and then they were summed together. And so you will see that areas which have unique importance or unique biodiversity from the chronogram perspective, that is, that have older lineages with small, and at the same time, those taxa have smaller ranges, were identified in that blue color. In areas that have more divergence in terms of mutation taking place using phylogram were highlighted in yellow. And we can see, for example, those areas include some relict untouched areas not south of San Francisco, for example. So even though we are found in California and there's a lot of built up areas in California, even some of the small green spaces we have left may contain a lot of biodiversity of significant evolutionary values that we cannot just replace them with some plants, invaded plants from other regions because and things like that. For example, bringing plants from Australia or wherever, because those areas may have a lot of important conservation, biodiversity of conservation value. And then areas in pink represent areas that where we have concentrations of lineages that have higher diversification or net diversification than expected. And then we see the union of all of those are represented in black. And so we can see from there that there are some portions of the North Central Sierra Nevada that are yet to be protected, which has a lot of important biodiversity there. And then, of course, areas northwest of that on the coastal parts of California, of Northern California, and then below in Port Point Conception, if I'm correct about that, and some other parts of Central California, which have unique biodiversity covering all of the three facets. And so let's look into some of those examples. For example, the North Central part of Sierra Nevada, particularly in the Sierra foothills, have been noted to have a lot of biodiversity, and these areas are pristine. Many of them, if I remember correctly, as on private lands, where they are owned by private landowners who have ranches, who bought up thousands and thousands of acres of land, but thankfully some of them are not heavily degraded, as you will imagine. We have some areas, in fact, especially in like Plumas County, where there's a lot of biodiversity. And funny enough, these areas in California maps shows that these areas, especially the north central part of California, have been poorly surveyed in terms of botanical collections. These areas have not yet been well studied, well collected, and in fact, there are several organizations, including the CMPS, that are funding collections, annual surveys, to be going to these areas to go and do more collections for plants in that region. And then we have Mount Armintel, which is not too far from us here, which is another area that has a lot of biodiversity in terms of a combination of concentrations of lineages that are older, small ranges, not yet protected, and also a lot of divergence and diversification in those lineages, which is begging for attention and protection. And then, of course, the Cape Mendocino, the Humboldt Bay, which is closer, which is in the north coastal ranges of California, which is really, really another important area that has a lot of biodiversity that is also needed for protection. I'm sure Brent will also be able to chime in at some point, showing some areas that are recently recommended for protection, where some organizations actually donated money to be able to protect those areas, which were also shown from this study that they have important biodiversity. So these are some of the conservation applications of this study, whereby we can model the phylogenetic diversity of different areas using different metrics given the facet of phylodiversity we choose to use. And so in this case, we used all three, and then we have a union of them, and then policymakers have a decision to like say, well, let's use divergence as the criteria to select new areas, or let's use diversification, or let's use older lineages, or let's use all of them. And so policymakers, conservation managers can have different options of what to choose, and then, of course, bringing in the principle of complementarity into it, such that you can make that decision that given the areas that are already protected, which areas need to be protected without having to be redundant in the protected areas that are being created. But beyond that, we could also use spatial phylogenetics to compare biodiversity patterns across taxonomic groups. This is something that Brent has already explained in response to someone's question, whereby we can compare phylodiversity patterns for plants, for seed plants, and compare that with butterflies. So you can compare between plants and bees. I know there are some group of experts that are currently working on creating a phylogeny of North American bees. And once that is available, we can compare that with that of North America of plants to see whether there's some level of coevolution going on there, whether there's some congruence or not. You can compare, and then also, in addition, we can relate phylodiversity patterns with different climatic conditions. For example, this is another question, another way of validating a response to an earlier question, whereby once you have this map, you can download the map, you can download the values for each of the grid cells of the map, and you can compare those with paleo or current or future climatic conditions to see which paleoclimatic conditions are associated or correlated with these biodiversity patterns that we are seeing, or is it temperature, is it precipitation and things like that? You could use that to answer various questions. Another way of looking at it is you can even integrate functional traits into the mix. For example, we could ask the question that given the lineages that are coexisting in a particular region, is there some ecological interactions that is driving or combining with evolutionary drivers to drive the biodiversity patterns we see in a particular grid cell? And this is something that Brent and I are currently working on at the moment, and we're really excited about, to see the role of functional traits in addition to the occurrence data to be able to assess phylogenetic patterns. And so these are several ways through which we can apply spatial phylogenetics to understanding the patterns of biodiversity on the landscape. And so we can end the chapter or this workshop at this point with these really exciting applications and many, many other areas that are currently being developed to apply spatial phylogenetics to explain biodiversity on the landscape. And perhaps maybe you have some ideas too, or other questions about how these methods can be used. And so we can use the remaining time to answer those questions or have general discussions on the application of spatial phylogenetics.